Interesting talk. So our next speaker is Dr. Garrison. He's currently a associate professor at Department of Pharmaceutical Science as well. His research interest is in design of radiotherapeutics and molecular imaging as well. Today, I'm looking forward to his talk. I'll probably let him introduce it.
All right, that's great. All right, thank you so much. So thank you to organizers for setting this up. It's a pleasure to talk to you today. Today I'm going to be talking about our work developing targeted radionuclide therapeutics. Specifically, I'll be using protease inhibitors to improve NTS1 targeted agents which can be used for a variety of cancers. So I do have a conflict of interest to disclose. My wife and I have started a company to help translate some of the drugs that we develop to the clinic. So be aware. All right, so brief review of radiopharmaceuticals and nuclear medicine in general. Radiopharmaceutical design, there's typically 4 components. You have the radioisotope, which can be diagnostic, therapeutic, or both. Chelator or prosthetic group, its job is essentially to hang on to the radio metal in vivo. You have a linker that connects the chelation system, prosthetic group, to your carrier. And as far as your carrier, it's going to be a targeting vector that targets some unique identifier for your tumor, whether it be on the tumor cells, such as a receptor, or it's in the tumor microenvironment. So these targeting vectors or carriers can be based upon low molecular weight agents, such as small molecules and peptides, and I'll be talking about small molecules today. But they can also be high molecular weight carriers, such as antibodies, which are probably the predominant, but they also can be polymeric systems as well, and other type of higher molecular weight constructs. So I don't care what you're talking about, but there are 2 main factors with regard to radiopharmaceutical design that tell you how good your agent is. And that's either target accumulation, how much you can get to the target, right, or the tumor in this case, and your non-target clearance, how well does it clear from the background. That's going to affect your imaging as well as your overall therapy. And together, we use these values as tumor to non-target ratios to judge the performance of our drugs. So for low molecular weight agents, it has some advantages relative to external beam radiation therapy. One is that it selectively targets the tumor based upon the molecular signature that we're targeting. 2, it has the potentials and emphasis there to deliver higher tumor radiation doses. Partly because you have a lot lower non-target toxicity because the material clears so well or so quickly after administration. And perhaps most importantly, it has the ability to target systemically, so you can go after metastatic disease. So low molecular weight agents have advantages relative to antibodies and other high molecular weight constructs in that they are able to target the tumors quickly and they leave or clear the body quickly as well. However, one of the detriments of low molecular weight radiotherapeutics has always been the residence time within the tumor. Because these low molecular weight agents have high metabolism, high diffusion characteristics, their residence time in the tumor can be relatively short. And what a low residence time ultimately results in a low radiation dose delivery, which obviously is going to impact therapeutic efficacy. And so this has been a hurdle for the development of low molecular weight radiotherapeutics for the clinic. And this is the clinical problem that we're trying to address. Before I tell you about our approach, I'll tell you a little bit about our target. So neurotensin receptor subtype 1, or NTSR1, is a G protein coupled receptors, or G protein coupled receptor. It's expressed on a variety of cancers. Pancreatic, colorectal, and prostate are probably the ones we're most interested in our laboratory. Upregulation of NTSR1 begins in the early phases of cancer and progresses or increases as the cancer progresses. And upregulation of NTSR1 correlates with increased migration, invasion, and overall aggressiveness. A variety of studies have examined using human biopsies, the NTSR1 expression in a variety of cancers. I've shown 3 here. As you can see, at least for the prostate and colorectal, where they've kind of stratified the data, you have strong and medium expression of the NTSR1 in those biopsies. And this represents a good patient population to go after if you're designing a targeted radionuclide therapeutic. So it's highly expressed, and that's exactly what you want to see. So we are not the first group to recognize the potential of targeting the NTSR1 for cancer. Recently, there's been a compound called 3BP227. I'll just refer to it as 3BP. It's been evaluated in humans in a relatively small cohort. I think it's around 8. A planar and SPECT CT image of the patient is shown here on the right. This is a pancreatic cancer patient, and you can see obviously uptake in metastatic lesions in that particular patient. So they did dosimetry based upon the imaging, and they found that the 3BP was able to generate similar radiation doses as compared to external beam radiation therapy. It also had lower non-target radiation toxicity. So it looked very, very appealing as a potential therapeutic. And so currently it's in phase 2 clinical trials. However, if you do look at the data, 3BP suffers from the same issues as most low microwave constructs. Its residence time in the tumor is relatively short. So to our approach of how we're trying to increase that residence time. So we're using specific class of cysteine proteases called cysteine cathepsins. So cysteine cathepsins are indolizable proteases that are primarily associated with protein turnover within the cell. And so in those indolizable compartments within the cell, these are expressed at very high concentrations, 1 millimolar. Some of these cysteine cathepsins are extracellularly expressed, but their extracellular activity is very low. And that's because they're tightly regulated, as you might imagine, by a variety of mechanisms. So over the decades, people have developed irreversible inhibitors. So what I mean by irreversible, once they bind, that's it. They completely shut down the inhibitor. So they've developed irreversible inhibitors such as E64. So E64 is shown here. It has an arginine-like amino acid next to a leucine, and then it has an epoxide ring. And so what happens is this E64 is able to bind to this binding pocket, and then the catalytic cysteine or thiol is able to open up that ring and form a carbon-sulfur bond. And once that happens, that's a permanent bond, and it's not going to become undone, so it's irreversible. So you essentially form what's called an addict. The inhibitor binds irreversibly to the cysteine protease. And so our overall idea is try to combine NTSR1 targeted drugs or receptor targeted drugs in general with this cysteine cathepsin inhibitor, or trapping agents as we call it. So mechanistically, I'll talk you through it here. So here we have our NTSR1 targeted drug. This is our targeting vector. Of course, we have a radionuclide attached. We've included in this little cartoon a trap. So this would be our E64-like inhibitor. So this would be able to bind to cysteine cathepsins. So the NTSR1 targeted drug would be able to bind to the receptor and then be escorted into the indolizable compartments, right? And those compartments, as I mentioned, are high concentrations of cysteine cathepsins. So you will be able to officially form a bond between the inhibitor or the E64 or the trapping agent, whatever you want to consider it, and the cysteine cathepsin. And then by doing that, you will form what we call a high molecular weight addict. So you're going from a drug that has a low molecular weight and has a relatively high diffusion rate from the tissue, and you're going to a high molecular weight addict, which has a much lower diffusion characteristic, right? And so by this, we expect to produce agents that have significantly higher long-term retention and is capable of delivering higher tumor radiation doses and improving clinical potential, not just for NTSR1 targeted drugs, but for a wide variety of receptor targeted drugs. So in this particular project, we decided to do a heads-up comparison between 3BP, which is our clinical control that's currently in clinical trials, phase 2, and then we made a derivative which is obviously very similar to that of 3BP, but we've included this E64 inhibitor component, okay? So this is called INTAT E64, but I'm just gonna refer to it as INTAT. So I'm not going to go through the synthesis or a lot of the in vitro characterization we do. We just don't really have the time, but I will show you some of the studies that we do that hopefully prove the point. So the INTAT is T and the 3BP is our control C. And so if you incubate INTAT with cathepsin B, which is a cysteine cathepsin, it's our model cysteine cathepsin. So if you incubate it with cathepsin B, you see that you form an adduct. Right? However, if you take C, which is our control, you see no adduct formation. So this demonstrates that the E64 is capable of binding to cathepsin B, to a cysteine cathepsin. We did the same thing with live cells. We incubated our compounds with live cells, and we see adduct formation in those live cells when we take a look at the cell lysates. However, if you look for the control, there are no adduct formation. If you add a NTSR1 blocking agent, so if you prevent the internalization of the drug, you see that you're able to completely inhibit the ability, or you completely inhibit adduct formation. And this points to the mechanism. In order for us to form adducts within the cell, the receptor has to be able to bring the drug in and escort it to the endolysosomal compartments. So if that process is blocked, no adduct formation will be completed. All right, so we next took a look at in vivo biodistribution of the agents using this HT29 xenograft model. So HT29 is a colon cancer cell line, and it is known to be NTSR1 expressing. So the solid columns are the INTAT, and the hollow columns are the 3BP. So this is the percent injected dose. This is the absolute uptake. So just looking at the blood, the muscle, and the bone, both of these agents clear effectively from those tissues. You do see uptake in the small intestines for both constructs, and that's because the intestines are NTSR1 expressing, but you also see a relatively rapid clearance over the 1 week that we take a look at them. But the highest uptake and for the INTAT retention you see is for the liver, the kidney, and the HT29 tumors, and so I'll focus on that. So if you take a look at the difference between the INTAT and the 3BP, what you see is pretty obviously is that the INTAT has a lot higher retention over time, right? So for the liver, essentially the same uptake at 4 hours, however, very different retention profiles when you look at over the course of 168 hours. And that's pretty much reflected for every single tissue you see here. And so we learned a couple lessons here, or this shows us a couple things here. First of all, that we believe that this retention is due to this adduct formation, which I showed you earlier. But it also shows that any tissue, any cell that takes up this material that has this adduct formation capability will be able to have increased retention due to adduct formation. It doesn't really matter what cell type it is. If it internalizes the drug, it will form these adducts and have higher retention. So something that needs to be considered when you're using this approach. So what we normally do, we normally don't look at data as a percent ID when we report them. We rather look at it as a percent injected dose per gram. So we usually account for the mass of the tissue by dividing it by the total weight, the percent ID by the total weight of the tissue. And this is a way of giving us concentrations. It also helps to normalize the tissue variability between the, or I should say normalize the tumor variability when you're working with these xenograft models, because the tumor variability can be quite large. And so the error bars go much, or get much lower when you normalize for mass. But at any rate, what you see here is you see, from a concentration standpoint, that the kidneys have a much higher concentration than we do the liver. And so what you can take from that is that the kidneys, if we're going to use INTAT, is going to most likely be our organ at risk. The tumors have the highest overall uptake, and as you go through time, through 24 hours, you can start seeing significant differences. But at 72 hours, you see a twofold difference in retention. By 3 hours, you're seeing a threefold, or I'm sorry, by 168 hours, you're seeing a threefold difference in retention. So to demonstrate that, in fact, we are forming these adducts in vivo, we've done a number of techniques. I'm just going to talk about one. We did this SDS page analysis. So we took the tumor, the liver, and the kidney tissues from the mice. We were then able to form lysates of those and run an SDS page gel ureography. So what we see is we do see adduct formation in those tissues. Again, those are the tissues that took up our material, which demonstrate that we are forming these macromolecular adducts. However, if you look at 3BP, we don't see any of those adduct formation. So with the help of Dr. Megan Hun from the University Department of Radiation Oncology, we did human dosimetry estimates using a Lambda EXM. We found that the NTAT was able to deliver a 2.7-fold higher dose, so 19.30 centigrade per millicurie relative to the 3BP, which is 7.26. However, because of the uptake and retention profile, we increased our overall kidney dose from 0.25 for 3BP to 2.88. Now, 2.88 is still relatively low relative to most targeted radionuclide therapeutics that are in the clinic. For example, Lutathera that's about in the same range of Lutathera. So this still is very reasonable for translation. Now, as far as the increased uptake, obviously the kidney is going to be your organ at risk most likely. Conservatively, when you take a look at the dose for the kidney, you're looking at around 23 gray limit as far as how much dose you want to deliver. However, there have been many papers recently that have questioned that as overly conservative, and you might be able to get away with a little bit higher. But nonetheless, the tumor to kidney dose ratios is around 6.7, which is very promising for translation. However, there are also ways to further improve the tumor to kidney ratios, and one of those is renal blocking. So generally, basic amino acids can be transfused along with the drug in order to increase or decrease the recognition and uptake in the kidneys, and that would be expected to further increase tumor to kidney dose ratios. So in summary, we've explored, we believe, a unique endolysosomal trapping approach to improve the retention of receptor-targeted constructs. This approach has led to a substantial increase in retention by forming these macromolecular adducts. We've actually applied this technique to a variety of receptor-targeted drug systems. It's worked in all of them. So we think this has a lot of potential to be applied to the entire field of receptor-targeted design. As far as NTAD E64 goes, it appears to be a very promising targeted radionuclide therapeutic. We're currently doing therapy studies right now. If successful, we aim to do necessary studies to obtain an exploratory IND to confirm the pharmacokinetics of the agents in humans. Of course, I'd like to acknowledge my group. Dr. Wei Fan and Dr. Wenting Zhang were the postdocs who did the great majority of this work. Of course, Dr. Megan Hun from UNMC Radiation Oncology for the human dosimetry studies, and of course, the funding and support. Thank you so much.
All right, thank you, Dr. Garrison. Okay, I'd like to introduce the next speaker, Dr. Christine Dickinson. She's an assistant professor from College of Nursing. Her research has been focused on the genetics and the mitochondrial profiles on cancer-related fatigue. So let's hear the talk on radiation-related fatigue. Thank you.
Thank you to the organizers and committee for the opportunity to present today. And what better time to talk about fatigue than Sunday afternoon at 4:30. So I have no conflict of interest to disclose, and I'm going to